So the reproductive systems are one of the few systems that are actually different between males and females, obviously because of the different roles that males and females play in reproduction. Fair warning here, we will be discussing the male reproductive system. You will probably see pictures of penises and, potentially vulva and other sexual organs. If you aren't okay with this, you may want to reconsider, whether anatomy and physiology is an appropriate class for you, but in any case, you have been warned. So, I want to start off by talking a little bit about the reproductive system in general. The reproductive system contains two parts, what's called the primary and secondary sexual organs. The primary sexual organs are the gonads. These are what produce gametes, or sex cells. In males, these are the testes. In females, these are the ovaries. Everything else is a secondary sex organ that's a part of the reproductive system. These are organs that could be involved in delivering gametes to their proper functional place, in nurturing a developing fetus, both inside and outside of the body, as well as conveying, I think I want to say sexual response and signaling sexual development. So these are not, these are things that are necessary for reproduction, or at least helpful for reproduction. In males, they consist of all of the ducts that deliver sperm, all of the glands that deliver hormones, the penis, which delivers sperm as well. In the females, it consists of uterine tubes, the uterus itself, the vagina, and the vagina, which receives sperm, and the uterus, where the developing fetus, develops. I want to focus this particular lecture on the male reproductive system. In general, the male reproductive system is less complicated than the female reproductive system. It has fewer functions, and its functions have somewhat less complicated regulation schemes. The main functions of the male reproductive system are, first, the production of sperm, which is a process called spermatogenesis. Secondly, the delivery of sperm to the female reproductive system. This process is typically called sex, and it ends with ejaculation. And third, the production of hormones that are going to regulate the development of the male reproductive system. The reproductive system is actually one of the few systems that does not really become fully developed until, after puberty. That's kind of the definition of what puberty is. So, male reproductive system anatomy, as you see here, this is all of the, or most of the main components of the male reproductive system. The primary reproductive organ, you see here, these are the testes. Sperm are made in the testes and come out through the vas deferens into the epididymis, which is this set of tubes next to the testes, and, all right, travel up the seminal tubules and the vas deferens, which is this here, is the vas deferens. If you have a vasectomy, that's what gets cut. Sperm then travel up and around and out here at the ampulla of the ductus deferens and into the prostate. The seminal vesicle also will secrete substance surrounding the sperm, and we'll talk about exactly what that is. And this joins in with secretions from the prostate that basically are ejected into the urethra, through the penis, and then out. And that's the path of the reproductive system in males. The penis itself consists mostly of spongy tissue, and I will describe that in a little bit, but first off I want to focus on the primary reproductive organ, the testes. So the testes in males consists of a set of glandular tissue that is covered by some connective tissue called the tunica albuginea. This is where the sperm gets made, and I'm going to go over the process of spermatogenesis in just a bit. The sperm, once made, is secreted into the epididymis. This is the, it's sort of like some squishy tubes off to the side of the testes that sort of wrap around inside it, and eventually the tail of the epididymis wraps into the vas deferens or the ductus deferens, which is what carries the sperm up to the prostate. The testes are attached to the body. I mean, they basically dangle there in the body. They're attached to the body through the spermatic cord. The spermatic cord contains the ductus deferens, as well as the testicular artery and vein, which deliver blood, and it is surrounded by a muscle called the cremaster muscle. Yes, I understand the jokes of the cremaster muscle. This spermatic cord is surrounded by a set of muscular and connective tissue called the connective tissue is called the tunica vaginalis, and the muscular tissue that surrounds the tunica vaginalis is the dartos muscle, right here, which is then surrounded by the skin of the scrotum. These two muscles, the cremaster muscle and the dartos muscle, collaborate to basically move the testicles around. The testicles are not self-cooling, and they have to actually be at a slightly cooler temperature than normal body temperature in order to produce sperm. Normal body temperature is 37 degrees Celsius. The testicles have to be at 35 degrees Celsius or below in order to effectively produce sperm. But if the temperature gets too cold, then they also can't produce sperm. It's kind of a Goldilocks zone, where they can't be too hot, they can't be too cold. That's the purpose of them dangling along down below like that. It's to keep them cool without getting too cool, right? So the cremaster muscle, basically what it does is it raises and lowers the testicles by, when it contracts, it pulls them up, and when it expands, it lets them go down. So it actually, they dangle from the spermatic cord, and it'll raise and lower them on the spermatic cord. The dartos muscle does not directly lift the testicles, but what it does is it causes the scrotal skin sac to either droop or become wrinkled, right? So in cold weather, the dartos, or any sort of cold thing, or also under the influence of adrenaline, by the way, I'll get to that in a second. But in cold weather, the dartos muscle will basically contract, and that will pull all of the scrotal sac sort of farther up into a little wrinkly body that presses the testicles further up close to the body and reduces the surface area in contact with the outside world, thus preserving body heat. A third mechanism for keeping the testicles the right temperature is that if they were directly nurtured by blood, blood from the body, that blood's coming out at normal body heat, which is too hot for the testicles to take. So you have warm blood coming down the veins, and you have cool blood, warm blood coming down the arteries, and you have cool blood coming up the veins. And what you do is you get a flow of heat from the warm blood coming in to the cool blood leaving. This means that the blood that enters back into the central body is warmer than it would otherwise be, and it means that the blood that is coming down to the testicles has cooled off a bit, so that it does not overheat the testicles. It should also be noted that under the influence of adrenaline, usually the cremaster and dartos muscles will contract and pull the testicles farther up, closer to the body. This is part of the fight or flight response. It gets them out of the way. Whether you're running or whether you're fighting, in either case, you don't really want the balls dangling around down there where they could get injured. Here you can see an internal section of the testes. And you can see that the testes here are divided into these lobules, and each of these lobules contains lots and lots and lots of squiggly little tubes. So each lobule contains a descending squiggly little tube and an ascending squiggly little tube that join together here in the rete testes. These lobules, these are where the sperm is made. And I'll talk about that on the next slide when I talk about the process of spermatogenesis. But the sperm that are made in these lobules are basically drain into the rete testes, and the rete testes drains through the efferent ductule into the epididymis. Follows down the tubes of the epididymis to the tail of the epididymis, and then up the ductus deferens or vas deferens, both are appropriate terms, and then back up into the body. So the process of spermatogenesis, this is taking place in those tubules that are in the lobules. Those are actually called the seminiferous tubules. Semen basically means sperm. Seminiferous means sperm-generating tubules. Remember that the process of making sperm, spermatogenesis, involves meiosis, creating gametes from usually stem cells, diploid cells, that are going to undergo meiosis to produce haploid gametes. The process starts here on the outer edge of the seminiferous tubule. Here you have a special type of cell called a type A spermatogonia. Type A spermatogonia you keep around, right? So hopefully you don't lose them through the course of the process of spermatogenesis. These cells are constantly undergoing mitosis. So when they split off, when they mitose, they divide into two cells, right? One of those cells remains a type A spermatogonia. The other cell goes up here and becomes a type B spermatogonia. So it reproduces, one remains a type A and stays at the edge of the tubule. The other moves a little bit further in and becomes a type B spermatogonia. This is still a diploid cell. Type B spermatogonia enlarge and develop along this path and become what is called a primary spermatocyte. That means a cell that's going to become sperm. It's the primary spermatocyte that undergoes meiosis. Specifically, a primary spermatocyte undergoes the first half of meiosis, meiosis 1. So this primary spermatocyte is a diploid cell. That's what this 2N here means. It's diploid. It undergoes meiosis 1, and meiosis 1 is where chromosome reduction takes place. So at the end of meiosis 1, you have two haploid cells that are both called secondary spermatocytes. These secondary spermatocytes undergo meiosis 2, each producing 4 spermatids. At that point, meiosis is complete. These are the cells that will go on to become sperm. But these spermatids are not yet sperm. They're haploid gamete cells, but they have to undergo a development process called spermiogenesis. The whole process is called spermatogenesis. This whole thing is spermatogenesis. This last portion of it is spermiogenesis, and that is the process by which this haploid gamete cell develops sperm-like characteristics. Sperm are basically little genetic payloads, right? All they need is, like, the DNA and a mechanism to get where they're going, which is to the egg. And that means that they're basically going to jettison everything else. Most of their cytoplasm gone. Most of their other organelles, gone. What they do is they keep a bunch of mitochondria back here, and they grow this long flagellar tail. The mitochondria are necessary to give it enough energy to swim quickly. And other than that, they've just basically got this genetic payload in the front. These have undergone spermiogenesis and have become spermatozoa. The spermatozoa, when they're produced, are let out into the lumen. That's the inside of the seminiferous tubule. They don't start off fully mature, even though they look, the spermatozoa looks mature. They actually don't start swimming and wriggling and become completely functional sperm until they move through the epididymis into the vas deferens. So we just described the process of, spermatogenesis, and the sperm from there get into the epididymis and into from there into the ductus or vas deferens. The vas deferens leads from the testicles up through the spermatic cord, over the bladder, and into the prostate. There are several spermatic ducts along the way here, all right? So, semen is composed of sperm, which are the cells, and seminal fluid. I'll get to exactly what the seminal fluid is made out of later on, but that seminal fluid basically consists of a bunch of stuff to help the sperm do their job. And it is made by a few different places. First off, so you can see here and here, those are the ampulla of the vas deferens. This is what's going to put the sperm into the prostate. But as the ampulla enters into the prostate, you see this gland here. This is called the seminal vesicle, and it makes part of the seminal fluid. The prostate gland here, this is where basically the vas deferens and seminal vesicle empty into the urethra. The prostate gland also makes some seminal fluid, a milky secretion that forms about 30% of semen. The mixture made by the seminal vesicle makes about 60% of semen. So as you can see, the sperm cells themselves are less than 10%. Those are the constituents of semen. This here is called the bulbourethral gland. It produces a not semen, but this like clear lubricating fluid that's made prior to ejaculation that basically allows the head of the penis to penetrate the vagina, or makes that a little bit easier. It also, the urethra, naturally, because urine passes through it, naturally the urethra is acidic. That level of acid would actually kill the sperm. So the secretion that's produced by the bulbourethral gland neutralizes the acid in the urethra, making it a place where sperm can live. In order for sex to occur, an erection usually takes place. So the mechanism of erection is contrary to popular belief. There are no bones or muscles in the penis. well, there's a few muscles, but they're mostly involved in moving the skin around and things like that. The penis is primarily made out of 3 columns of spongy tissue. Basically, this is tissue that's composed of a bunch of capillaries for blood. And what happens when the penis becomes erect is it basically fills with fluid. Specifically, it fills with blood. Under the influence of the parasympathetic nervous system, remember that's the rest and digest nervous system, the neurotransmitters acetylcholine and nitric oxide, that's NO, or laughing gas, are secreted. These are both potent vasodilators. So what happens is, under the influence of the parasympathetic nervous system, these are secreted. They're vasodilators that allow more blood to enter the penis. That blood is entering through the deep artery that's found in these 2 what are called corpus cavernosum. Those are the primary erectile tissue of the penis. This third area here is the corpus spongiosum. That's the chamber through which the urethra passes. Basically, the sponge around here serves as a support structure so that during erection, the urethra doesn't close off and prevent ejaculation. These 2 areas, the corpus cavernosum, those are the areas that become erect. They fill up with fluid, primarily blood, and that is what causes the erection. The process of ejaculation is actually a combination of the sympathetic and parasympathetic nervous systems. Remember that in order for erection to be maintained, the parasympathetic nervous system has to be activated. So that's the first start, is the parasympathetic nervous system is activated, allowing fluid to enter into the penis. Stimulation, either physical stimulation of the genital region or potentially mental, visual, or other stimulation in the brain. The brain is actually one of the major sex organs, causes increasing stimulation of the sympathetic nervous system. So you've got the parasympathetic active, and then the sympathetic nervous system rises through a process called excitement. As this process happens, so you can see here, the pelvic nerve is sending parasympathetic nervous systems to allow erection, and during orgasm and ejaculation, sympathetic nervous system basically becomes more and more and more active until it effectively overcomes the parasympathetic nervous system, until it becomes more active than the sympathetic nervous system. This causes the seminal vesicles and the prostate to secrete the components of seminal fluid. The semen, the cells, and the seminal fluid gathers in the urethra. This sends signals to the spinal cord, which reflex short circuit. Those are afferent signals that say, hey, the prostate is now full of semen. That sends efferent signals to the prostate, and also to a somatic muscle called the bulbocavernous muscle, which causes basically the bulbocavernous muscle squeezes around the prostate and around the urethra, and that squeezing process is what causes ejaculation, shooting the sperm out. Post this, the sympathetic signals are stronger, and the parasympathetic signals are weaker, meaning that the artery constricts, reducing blood flow into the penis. Typically, erection is then lost, and the penis becomes flaccid. That's what happens with male ejaculation. Or at least that's what happens mechanically with male ejaculation. There's also a bunch of stuff going on in the brain, but it's not central to the reproductive system at the moment. Semen itself usually consists of 2 to 5 milliliters of fluid. 60% of it is seminal vesicle fluid, 30% is prostatic fluid, and 10% is sperm and secretions of the spermatic duct. In a normally fertile male, the sperm count is 50 to 120 million sperm per milliliter. Generally, if you are at half of that minimum normal value, 20 to 25 million sperm per milliliter, this is sufficient to produce infertility. This doesn't necessarily mean that it's impossible for a male with a low sperm count to impregnate a female, but it reduces the likelihood dramatically. So often, assistance is basically required, and there are things that can be done. You can concentrate the sperm artificially and then undergo, artificial insemination or something like that. And it is possible that natural fertilization can occur, but it makes it unlikely to the point where throughout the course of a normal relationship we would not expect fertilization to occur. The prostatic seminal fluid contains calcium citrate phosphate, a clotting enzyme, and a protein hydrolyzing enzyme called a serine protease. That will become important in a second. The vesicular seminal fluid is thick and viscous, and it contains fructose, which is a sugar, as well as other carbohydrates, again citrate, as well as prostaglandins, which are not exactly hormones, but very similar to hormones, and a protein called proseminogelin, sorry. Why have all this stuff? What's the purpose of all of these things? Why not just have the sperm floating in, I don't know, water, or even blood, or something like that? There are actually a number of important things that the seminal fluid, important properties that the seminal fluid plays. First off, stickiness, right? So the clotting enzyme, which we talked about, which was a serine protease, this clotting enzyme, when it is mixed with that protein, proseminogelin, right, it will catalytically work on it and turn it into seminogelin. Usually this pro prefix means that it's an inactive precursor, right? It's something that's made in an inactive form because you don't want it to start working inside of the penis, but is then converted into an active form when it's mixed with something special. That would be the clotting enzyme. This is actually similar to the way your blood clotting works, although it has a slightly different purpose. What this does is it makes the sperm sticky, and it makes it clot up and stick to the inner wall of the vagina and the cervix. This makes it less likely that the sperm is just going to drain out of the vagina. Make sure that it sticks to the walls and then heads where it's supposed to go, which is into the cervix. These clots are semi-solid and will be taken up through contractions in the uterus. It's thought that female orgasm plays a role in promoting the uptake of these seminal clots into the uterus, although that isn't well understood. To be entirely honest, for various reasons, male orgasm and ejaculation has been studied a lot more than female orgasm has. And scientists still don't have a complete understanding over what the evolutionary role of the female orgasm is. But it's thought that, at least in part, it plays a role in promoting uptake of these clots into the uterus. About 20 to 30 minutes after ejaculation, the clot basically melts, allowing the sperm to continue its pathway once it's been taken up into the uterus. The second main property of semen is to promote motility and movement of the sperm. Specifically, the citrate and phosphate and calcium will raise the pH of potentially the urethra, the male urethra, but also of the vagina. The vagina is usually an acidic area. That acidic environment is hostile to the presence of sperm. It would kill off the sperm. And so these components will raise the pH from 3.5 to 7.5. 7.5 is a neutral pH, which is a fine environment for the sperm to survive in. The fructose and sugars basically serve as a food source for the sperm. They've got a long trip ahead of them, and they are going to need some energy for the road. It provides a power source while they are basically swimming to the ovum, or at least in search of the ovum. The prostaglandins, like I said, they're not hormones technically for a number of reasons, but one is that they act in a different person than made them. But they serve as a chemical signal that thins the mucus of the cervical canal and stimulates contractions in the uterus to basically spread the semen around so that it has a greater likelihood of finding an egg that is hanging around in there somewhere, or finding their way up to close to the ovary. And actually, most eggs are fertilized well before they get to the uterus. Our last function of the male reproductive system is hormonal control of the male reproductive system, and also of puberty and pubescent development. The hormonal regulation here is complex, although not anywhere near as complex as it is in the female reproductive system. Let's see here. Basically, starting from the hypothalamus up here. The hypothalamus releases gonadotropin-releasing hormone into the anterior pituitary, which we've already studied. That stimulates the anterior pituitary to produce follicle-stimulating hormone and luteinizing hormone. In males, follicle-stimulating hormone acts on the Sertoli cells in the testes to secrete a protein called androgen binding protein. Luteinizing hormone, on the other hand, acts on cells called Leydig cells in the testes, and that stimulates them to produce testosterone. Testosterone is the main hormone controlling the male reproductive system, or at least the later parts of it, and is the main androgenizing hormone. In the presence of ABP, so if you have testosterone plus androgen binding protein, those two things together, that tells the body to stimulate spermatogenesis and to make sperm. So that only happens if you have both luteinizing hormone and follicle-stimulating hormone. You require both of those to make sperm. Testosterone has other effects, however. First off, testosterone stimulates the libido in the hypothalamus and the thalamus and other parts of the brain. Testosterone is responsible for development of secondary sex organs, as well as secondary sex characteristics, which are things that aren't organs exactly, but are still sexual signifiers. Things like the development of changes to the larynx that cause the voice to deepen, increasing muscle density and bone density, stimulating the growth of pubic hair and other body hair in males. Those are all secondary sexual characteristics, as well as stimulating the development of the penis and the testes during puberty. Testosterone has an inhibitory effect. This is through negative feedback on both the hypothalamus and the pituitary. Basically, the more testosterone you have, the less the pituitary secretes FSH and LH. And that makes sense, right? You don't want your testosterone to just keep rising more and more and more and more and more. So once you've got enough testosterone there, it turns off the pituitary and the hypothalamus. They stop secreting their hormones, the Leydig cells stop secreting testosterone. Eventually, testosterone levels go down, and the hypothalamus and the pituitary become active again, telling you to make more testosterone. Basically keeping the levels of testosterone at a constant medium-high level. It's different in every person, but in most males of reproductive age, it's medium-high. It does decline over time. One other important feedback mechanism is these Sertoli cells, which are activated by follicle-stimulating hormone to generate ABP. They can also produce a hormone called inhibin. Inhibin inhibits follicle-stimulating hormone. So, right there. It inhibits follicle-stimulating hormone, but not luteinizing hormone. So it will not inhibit the production of testosterone, but it will inhibit ABP, and thus will inhibit spermatogenesis. So it can reduce sperm production. So inhibin reduces sperm production without reducing testosterone. And that's an important distinction. The main effects of testosterone on the body. So in a developing fetus, and we'll talk about this further in class, but in a developing fetus, once the testes are first made in the first, towards the end of the first trimester, the testes begin to secrete testosterone. That testosterone is what causes the fetus to develop as a male. Without that testosterone, the fetus will develop as a female fetus, even though it has testes. This process is called masculinization. Once the fetus is born, usually the secretion of testosterone stops, or at least goes way down, until puberty. In puberty, the secretion of testosterone resumes, allowing sperm production and causing the development of those secondary sexual characteristics that we talked about. Increased muscle growth, increased thickening of vocal cords to deepen the voice, thickening of bone, growth of body hair, and, as all of us males remember, increased activity of the sebaceous glands, causing you to secrete lots of oily skin and have lots of acne. Well, I hope that this has enlightened some aspects of the male reproductive system for you all. And my next video will concern the female reproductive system, which is a little bit more complicated because of its roles.